Hi everyone, my name is Christin Tran, and I'm a senior at Georgia State University conducting organic synthesis research under Dr. Swazette Mooring's laboratory. My project is on synthesizing asymmetrical CXCR4 modulators. Chemokines are a large family of secreted small proteins belonging to the cytokine superfamily that mediate cellular activation, differentiation, and trafficking. Specifically, CXCR4 belongs to one of the four chemokine receptor families, CXCR. CXCR4 is a 7-transmembrane alpha helix G protein coupled receptor. CXCR4 is involved with healthy physiological cell functions such as triggering stem cell differentiation to repair tissue damage, migration of narrow crest cells for normal brain development, and induction of embryonic stem cells for cardiogenesis. However, when bound to its single endogenous ligand, CXCL12, the binding interaction of the receptor and ligand will mediate cell chemotaxis, a pathway that we can use for cancer metastasis. Therefore, as CXCR4 binds to its different ligands, a signal is triggered. In addition to its natural ligand, there are viral ligands like HIV-1 and synthetic ligands like AMD 3100 and the compounds I am currently synthesizing. It's important that once these synthetic ligands bind, the antagonists block the disease and drug-related pathways, but still allow normal physiological functions to occur. Our main focus is to block the disease pathway for cancer cell metastasis and inflammation. AMD 3100 is highly potent as a CXCR4 antagonist, but lacks oral bioavailability and must be administered by intravenous infusion, which limits its clinical applicability. AMD 3100 is also only approved to be used for stem cell mobilization instead of as an antagonist. New compounds with better efficacy are necessary due to the cardiotoxicity and poor oral bioavailability of AMD 3100. WZ811 was synthesized to combat these problems, but ultimately it also lacked oral bioavailability itself. This slide shows the progression of synthesis of synthetic ligands such as AMD 3100. From AMD 3100, the basic structure is improved by modifying the two cyclone rings into a dicyclone macrocyclic N-pyridinylmethylene molecule as a way to reduce the basic amine groups attached. This is for therapeutic purposes. To increase efficacy and oral bioavailability, the synthesis of better CXCR4 antagonists continues. In Dr. Mooring's lab, we are using the progression of these precursor molecules to guide our strategy of synthetic design. Current research within our group has focused on designing and synthesizing analogs based on WZ811 by altering the phenyl ring with other heterocyclic or aromatic rings such as thiophene and pyridine. Previously, part of Dr. Mooring's lab has synthesized symmetrical analogs such as 2,5-thiophene, especially the symmetrical substituents 4-methoxyaniline, 2-methylaniline, and 2-chloroaniline exhibited above 60% inhibition in the biological test. Up to now, all our molecules have been symmetrical, meaning that there are the same substituents on both sides. Here is where my work begins to create an asymmetrical compound. Our first goal was to synthesize the precursor for the overall synthesis of our target compounds. My work involves creating an asymmetrical molecule with a different amine on each side. The substituent amines were chosen based on the results from Dr. Teresa Gaines and Francisco Garcia's synthesized analogs. Substituents with high affinity in the binding assay and demonstrate a reduction in inflammation during the polyedema test are used. For the synthesis portion, the central thiophene ring is modified through stepwise reactions leading to two different functional group attachments using reductive amination. In this reaction scheme, in order to have an asymmetrical molecule, I protected one side so that there's only one other place for a substituent to attach to. One equivalence of ethylene glycol is the most optimized amount that I found to yield a monoprotected aldehyde. After the protection, a reductive amination with 4-methoxyaniline was performed on the unprotected aldehyde using sodium cyanoborohydride. After the reductive amination, deprotection of the ethylene glycol is performed with cerium ammonium nitrate. With the last and final step, another reductive amination with 2-methylaniline is performed. Mass spectroscopy and NMR spectra were obtained at each step for verification of the compound. I am currently on the last and final step. The methods to synthesize the precursor were modified continuously in order to synthesize our target compounds and maximize yield of products. The first 3 steps to synthesize the precursor compound were successful. In the fourth and final step, another reductive amination can take place for the second aniline attachment. The final asymmetrical compounds will be sent off to Emory University School of Medicine, Dr. Shim's lab, to test their efficacy as CXCR4 modulators using biological tests such as the Paul edema test and matrigel binding assay. This thiophene-based model will be extended to pyridine-based compounds. I would like to thank Dr. Suzette Mooring for her bountiful mentoring and support. Thank you for listening to my poster presentation. If you have any questions, please let me know.